Hello, my name is Hunter Gale, and welcome to the Soil Resource as a Basis for Life. Don't forget to use the chat feature on the right side of the screen. If you have questions or comments as we go along, please feel free to add them there. At the end, we will take a few minutes to answer some of your questions. Now, I'd like to welcome Dr. Robert Kremer to present the Soil Resource as a Basis for Life. Dr. Robert Kremer holds a BS and MS in agronomy from the University of Missouri and PhD in soil science and biochemistry from Mississippi State University. He is currently a consulting soil scientist and holds courtesy appointments as adjunct professor of soil science at the University of Missouri. Go Tigers. In 2014, he retired after a 32-year career as a microbiologist with the USDA Agricultural Research Service. He is a prolific author, having co-authored 2 books, 30 book chapters, and authored or co-authored 165 refereed research articles. He began studying soil health in the early 90s and was among the first to show the association of microbial activity with soil health in various agro-ecosystems. Using biological indicators, his studies demonstrate that good soil health is associated with sustainable agricultural practices. Dr. Kremer's main objective is to move farming toward reduced soil disturbance and fewer chemical inputs that degrade the environment. That being said, Dr. Kremer, please take it away.
Thank you, Hunter. And I'd like to thank all the organizers and the staff of this conference for inviting me to do this. So the idea was to give a review on the soil resource as it is related to plant growth, particularly in native prairies or prairie restorations and as well as sustainable agro-ecosystems. So some of this may be a little bit of a review for those of you who are professionals in the field. So I hope I don't make it too simple, but I will go through some of the basics of soil science, and this should provide a good basis for the subsequent soil health presentation. So we'll just get right into it. What is it about soils? And I've got a few of the listings here, some of the ecological functions that really we hadn't really thought about for the past until maybe about 100 years ago, when we started really realizing what this body on the surface of the earth does. So, habitat provision for plants and animals, regulates our water, it through filtering through the organic and inorganic matter, production of food, feed, and biomass. And so the essential ecosystem services it mediates are things like flood regulation because of good water infiltration, vegetation attenuates runoff, and human survival. We often forget about how basic soils is to human survival when you think about food production, food security, and the replenishment and purification of drinking water. And I think we got to remember that only about 34% of the Earth's land surface is suitable for agricultural purposes. So we really need to protect it. And as the UN director says, multiple roles of soils often go unnoticed because they are our silent ally in food production. In contrast, soils have been relatively poorly managed in many global regions, including North America. And sustainable land management should be promoted to save not only soils, but our environments. Much like an old soils professor of mine said, we still treat our soils like dirt, and why do we do that? So let's get into, what soils are. It's taken, as I said, some time for a consensus to come up with to define what it is. And one of the earliest definitions was by Hans Jenny, who was a very famous soil scientist and was stationed at the University of Missouri for several years back in the 30s and 40s. But he found that soils are a dynamic natural body. That's the first time that was really recognized, that it was a natural body rather than just a consolidated mass substances on the crust, on the surface of the earth. But they form due to combined factors of climate, biotic factors, activities, topography, parent material, and time. And several other people have expounded on that, saying that that it's characterized by horizons and a soil profile. And I'll show you that in a bit. And one of my other professors at the University of Missouri came up with it that it was a system. You know, it's a system that in which the energy of the sun, the atmospheric um, gases, and the biosphere, in other words, the recognition of the living organisms within the soil, interact within that landmass. And in the corner, I show a profile, a vertical section of a soil. This is a very nice looking soil, dark soil that you would find in North Missouri, Iowa, Minnesota. That is a very productive soil until it is subjected to certain management situations. So in reviews, valuables are a non-renewable resource for sustainable ecosystems. By non-renewable, I mean is if the improper management is imposed on a soil and the topsoil is ruined, it's very difficult, if not impossible, to replace that, especially in a human's lifetime. Those soils have been developed over tens of thousands of years, and once we lose the topsoil due to erosion, it's hardly you can never bring it back to the natural state that it was in. The quality and health of soils are linked to global food and water security. There are important implications for energy security, mitigation of climate change, degradation of soils due to land use changes, erosion, compaction, pesticide use, and so there is an urgent need to sustain these ecosystem services. So here's an example. This picture was taken not long ago, around 2010 to 15. Just gives you an idea of what some degraded soils look like. This is a landscape that was intensively farmed in southeast part of the U.S. for over 150 continuous years of cultivation, and it was not taken care of. And you have this erosion going on that gives you the gullies out here in the mass. Hardly anything grows on it but scrub pine, and this soil has been ruined. It's hardly it would take a lot to remediate it to grow anything at this point. And we have a lot of these areas throughout throughout North America. Here's a study that was just reported this year about a survey that was taken on erosion of soil within the upper Midwestern part of the United States. And what it shows you here, this is where the man is standing, uh, is the original prairie soil as the European settlers found it. It has never been cultivated. Uh, to the left of that, where the meter stick is a crop field that has had 160 years of cultivation, and this is in Iowa. And so you see there's been a loss of almost a meter of soil in that time. It's very deep soil. Um, and what was found is that they found that the surface thickness of soil is reduced 2 millimeters a year. Now, 2 millimeters is very low. It's about the thickness of a dime. But when you put that over an acre, 43,560 square feet, that can amount to 3 tons per acre per year. We're still having this problem in the Midwestern part of the United States. And worldwide, it's claimed that 40 billion tons of topsoil is lost annually. And we see it in our own state here. Here, this is in the Missouri River Hills near Glasgow, Missouri. Soybeans planted on a hilly landscape. And what do you see through here? If you look at the darker lines going down the hill slope, those are erosion gullies. There on the right is an open gully going through a cultivated field. The lower picture depicts corn growing in a river bottom, the Gasconade River in central Missouri. And what you see at the bottom here, see where those dark spots are, that is the river bank. And the dark spots is where the soil has sloughed into the river over time. This particular farmer is farming right up to the riverbank with no buffer uh, at all to establish perennial grasses to hold that soil back. So we still have this. We need to be educated to use better management practices that conserve these soils. All right, so let's get into some of the basics of soil, just to kind of back, you know, take a step back and review of what we know about soils. And if we know these properties, we are better prepared to implement good management practices to conserve the soils that are being used for food production, animal production. So soils are a four-phase system. Um, it's 50% solid, which constitutes minerals and organic matter. It's about 50% pore space, which means the spaces in between these particles that will be filled with either liquids or gases, the atmospheric gases. And then the fourth phase is the living and metabolizing microbiota, or the microorganisms of the microbiome that is living in the soil. It may only be 1% of that whole living biomass, but it's probably the most important and dynamic sector of the soil. And these interactions between these phases exist as a complex of chemical, physical, biological interfaces, and these all work together. When they work together, it may have a greater influence on the microbes and their activities than each factor by themselves. And just to depict this, in a pictorial, uh, the properties of soil are the physical, chemical, and biological properties, and they interact, and they all contribute to this fact component of organic matter, which is shown in the red overlap there. And that organic matter is a very key indicator on the condition of soil and on soil health, because it is not only formed due to the physical, chemical, and biological processes, but it also contributes to how each of those processes function. And we'll get more into that in a little bit. Relative, the soils have different particle sizes, and this is just a very simple diagram showing you the coarse sand at the top, how its size is relative to the finer sand, the circle there where you see the microorganisms, then very fine sand, and then you have the silt, which is that small circle towards the left-hand side of the diagram, and then very much below there's the tiny, tiny particles of clay. And clay is probably is the most important solid when it comes to chemical reactions, because of its great amount of surface area where all the metabolism and chemical reactions occur. These microbes that were shown here are obviously very much out of proportion. Some of those may be a little bit larger than the clay, but it just gives you an idea of what's in there: the bacteria, the fungi, the protozoa, and many other organisms that we'll talk about a little bit later. So each soil is characterized by its texture. Perhaps you've heard of silt loams, sandy clays, and that's due to the proportion of these particles that is in the soil. So we use what's called a textural triangle to determine what that texture is, and each um, axis represents, for instance, clay or silt or sand. And in order to determine a texture, sometimes it's used, using a physical instrumentation, or some people are very adept at feeling it with their fingers and getting the amount of they can figure out the amount of clay and sand in there and come up with the actual texture. But here, for instance, I have in the red lines, I've drawn from 20% clay across to meet about 60% silt and 20% sand. That would be the approximate proportion of a silt loam. And this silt loams are probably the most dominant textures, for instance, in Missouri. And the other thing to know about silt loams is that it is very um, prone to erosion. It will, erode off the surface very easily. So this is a lot of information here just to give you an idea of what the sizes of these components are and some of their importance. And as you can see here, the clay content contribute to many of these chemical properties. I don't really have a lot of time to go into it. One of the things I forgot to mention is that I taught soil microbiology for many years at the university, and that's a class where you get a broad range of students, and many students would know their soils if they were in agriculture, but they wouldn't know anything about microbiology. Many of the people, the biology people, would come in and they wouldn't know anything about soil. So, I had to come up and develop a very simple introduction to soil, so we try to get everybody on the same page, and did the same thing for microbiology. So you see these a little bit simplistic. That's kind of the reason why it's kind of taken out of my soil microbiology lecture. Well, anyway, to get back to this, the clay again contributes to a lot of these different chemical and biochemical reaction. Sand is most contributes most to the water and air content and movement. Contributes to these pore spaces. Silt is somewhere in between. We talk a little bit about pore spaces that are made by aggregates, wormholes, root channels, that kind of thing. Pore spaces is very important because that does contribute to aeration and water infiltration. And we'll get into that in a little bit. The structure of soil is very important because a well-structured soil will resist water erosion and will provide a good habitat for plant rooting structures. And soil structure is based on the aggregation of all those soil particles in organic matter that we just showed you in the previous slides. Um, the soil structure is stable because because it has an effect on the microbial activity. Microbes use their gums and polysaccharides to stick these particles together, make good aggregation, and provide an overall soil structure. The pores within the structure provide distinct microhabitats for the functioning microorganisms. So the importance of soil structure, as I mentioned, is aeration, water movement, and retention. In other words, water storage, and to facilitate the interaction between the biological components, which are mainly the microorganisms and the plant roots. Now, this is a depiction of a hypothetical soil aggregate. Um, what we see in the lower half here are all these particles together, primarily the sand. If you look in this inset here down here, it will point out the very tiny clay particles that are in there. And this organic material, the green substance, are all the slimes, or the gums, the microbial produced substances that are often called biofilms, similar to the tartar that you find on people's teeth. Those are biofilms. And what they do, they stick all these components together and they make that aggregate, part of the aggregate. And as you can see, this aggregate also has air spaces or pores in it. As you can see here where the nematode is, the green nematode towards the lower part of that diagram. And this is where it has the water and air, which is very um, necessary for life in this soil. You got to have these habitats, and this kind of depicts it very well. So we have this nematode up here. We have a mite. These filaments on either side of the aggregate that you see here actually serve to help additionally to strengthen that aggregate by binding them in there with these filaments that are binding the aggregate, as well as the root that you see that goes down on the right side of that diagram that further stabilizes this aggregate. And the microhabitat of the root is known as the rhizosphere, and that's where a lot of action happens from a metabolic standpoint with the microorganisms. So finally, not to get overbearing here, but the stability of the aggregate is a very important property. And this little experiment here shows that when we have a good soil with a lot of microorganisms, if you see on the left half of this depiction, the upper petri dish there shows aggregates that were sieved out of the soil and put here, and then it was subjected to rain drops of water to simulate raindrops over time. And then the lower left-hand corner, you see these aggregates are still there. They didn't fall apart, so they were very stable because they had that association with biology. And if you look on the right-hand side of that picture, you have these non-amended aggregates that were just sieved out of the soil. That didn't have a whole lot of plants growing in it. That was somewhat sterile. You hit it with water and they just fall apart. And you have no stability, no water holding capacity, no aeration. And this is oftentimes what you will see on some of these cultivated soils around the country. After a large rainstorm, it will just seal over. And once that seals, because the aggregates fell apart, then you begin to get runoff. It's not infiltrating into the soil. And this is a diagram of porosity, which is the soil volume that does not have solids in it. If you look on the right section, I'm sorry, the left-hand section of this graph, you see the square figures. This is the porosity under a corn production with no cover crops in between seasons. It just lays fallow. Low amount of porosity into the upper 10 centimeters, the upper 4 centimeters of the soil. Whereas when you're looking at something that's under on the left-hand side or right-hand side, which is under perennial grasses such as switchgrass, or even miscanthus, and cover crops, you have much more porosity there that's created by the root structure and the microorganisms, the earthworms in that soil. The porosity is much higher than the cultivated soil. And what does that mean? If you look at the text, improved aeration and water infiltration and water storage, reduced water runoff and erosion. So this is a good diagram of the benefits of using continuous roots in the soil, continuous vegetation on the surface of the soil. All right, so I've talked a lot about how important organic matter is. And to me, that is a key biological, well, actually, it's a chemical property too, because it is part of the chemistry of the soil, but it is a consequence of biological activity. And we can now look at soil organic matter components instead of looking at it as a soil organic matter content that one would see on a soil test report. These soil organic matter components begin with fresh residues on the soil. So grass clippings, crop residues, even leaf litter and this kind of thing that we see here depicted as plant residues in the right half of this diagram. It all starts with that and then is subjected to decomposition over time. And so what I'm showing here with this large arrow are the different types of microorganisms that are necessary to break down plant residues into a final portion of what we call soil organic matter in that lower part of the diagram, the dark material that is remaining in the soil. I show that this involves microbial consortia, which means a diversity of microbes working together, because one microorganism does not do this. It starts with a fungus, and then as this material, say this is straw or corn stalks, are broken down over time, then you have another microorganism group that comes in and begins breaking down it further. So these things are just torn apart and subjected to enzymes by these different organisms. And what's going on during that also is that any nutrients that are in the residue, such as nitrogen, phosphorus, sulfur, potassium is released by the microbes, and it's freely taken up by the microbes and by plants. So this is an idea of recycling this material so that it is useful for continued biological growth. The other thing I want to point out is this amount of carbon dioxide down here in the lower right-hand corner. When you have a metabolic process such as decomposition, there is an amount of carbon dioxide released because it is it's respiratory process. And it may be up to about 2/3 of the original carbon that is in this material is released as carbon dioxide. It's a fact of life. So this is how carbon dioxide is released again. It can be used by plants, which it comes back through the soil, or it goes into the atmosphere. But it gives you an idea that another reason why you just can't add compost to your garden or field um, in 1 year and expect that to be good for several years, because you're going to lose an amount of carbon dioxide every year. So it's good to keep adding that organic material on an annual basis. All right, so if we go back to the circular diagram, the components in the soil, we have an amount of fresh residue that is there, including this box of soluble organic carbon, which often is released through the roots of the plants. Um, and so that is immediately available to the microorganisms. This is what feeds them and keeps them living to decompose the material that goes into the soil. And then in the purple portion here, we have what's called the actively decomposing organic matter. We call this the active fraction. It is the most important fraction of carbon um, going into the soil because it is supporting the microbial activity. And it is also contributing to um, soil aggregation. That amount of organic matter can also glue the soil particles together, but it is also acting as a source of food for the microbes, and it's acting as a source of nutrients, as I mentioned before, that is for both the microorganisms and for plant uptake. In the yellow portion, there are many substances in the organic material, the plant residues, that are very resistant decomposition, such as the lignins and the tannins and those sorts of complex carbon compounds. Those eventually are broken down to a point, but then they become a part of the stabilized organic matter, or you may know it as humus. That may be up to 33 to 50% of that material going in there. But it is very important because what it does, it it adds stability to the soil. It is also can exchange and hold nutrients for plants rather than letting it be released into the soil water and then being leached away. It can it is also sorption material for pesticides and these kinds of things that will also hold it, and it gives the soil um, a structure that allows it to hold water and be aerated. And I'll get into that in just a minute. And finally, about 5% of the organic matter is living organisms. Those are the organisms that are working, but also these organisms are a source of nutrients as well. Because if you think about some of these organisms, their lifespans are probably 24 to 48 hours, and then they die, and then they become a source of organic matter or a source of nutrition for other living bacteria and fungi and other microorganisms, and they themselves are recycled. They are maybe 50% nitrogen. So there's a lot, it's a good nitrogen source that are in the bodies of these microorganisms that can be decomposed, and that material then can be released and add to the nutrients capacity of the soil and serve as a source for these microorganisms. Okay, so this gives you an example of the importance from a physical standpoint of that stored um, carbon in the soil that is part of the stable organic matter. This is a very uh, highly magnified piece of organic matter. And what is it, if it looks like to you on the left-hand side of the screen there, if it looks like a sponge, that's exactly what it's doing. It will absorb a lot of water, increases the water holding capacity of the soil. It allows better stress tolerance in for overall soil biology. And the graph shows, if you look at the x-axis, the amount of organic matter in the soil, and then the y-axis shows the percentage of water held. You can see there's a direct relationship. If you look at that upper curve, the field capacity or the water holding capacity of the soil increases tremendously with just a few percent increase in soil organic matter shown on the x-axis. So this is a very um, impressive type of uh, um, depiction of the importance of organic matter from a physical standpoint. Soil organic matter is once the soil is degraded, takes a very long time to rebuild that. If any of you were on the field trip the other day to Prairie Birthday Farm, I'd been working out there with Linda Hetzel for almost for almost 20 years, and we've been taking samples of her restored or reconstructed prairie about every year or every other year and analyzing it for soil components. And this shows the soil organic buildup in that reconstructed prairie over the last 15 to 20 years. Now, when she began reconstructing that prairie in the 90 mid to late 90s, that was on a farm that had completely degraded uh, the hill slope where this prairie now is located. There was probably 1 to 2 inches of topsoil left. So that meant there was hardly any organic matter. At this point, when we came in around 2002 or 2003, there was only about 2% organic matter. And a soil like that on the Missouri River Hills should have upwards of 4 to 5%, at least under a cultivated soil. So she established these native prairie plants, which I show a depiction of a profile here from another site of the deeply rooted soil roots in that soil up to 2 to 3 feet, and that is contributing to organic matter. And then you can see over time, as that those prairie plants were allowed to continue to grow, develop their root systems, we're getting upwards of over 6% organic matter in a span of 15 years, which is not bad. That's about 2% every couple of years, which would be a normal um, expectation on this soil since it was really degraded to start with. Sometimes you hear of folks that will say, "Oh, you can increase organic matter 1 to 2% a year." That's a very unique situation and not typical. Okay, so we've been talking about soils and their um, some of the basic properties. Those of you experienced in taxonomy of insects and flowers and plants uh, use a soil taxonomy system I'm sorry, a taxonomic system to identify those biological components. There is also a taxonomic system used for soil in soil genesis that we'll get to in a little bit, but it's based on these soil forming factors that was laid out by Yanni 80 years ago. And these factors are the climate, the living organisms, the parent material that it was developed in, the topography and time. And I just want to go over that a little bit because certainly I am not a soil classifier, but it's good to know about these because what this will tell you is that why soils are different on different landscapes, different regions where you go, and that management has to be aligned with the type of soil that you're working with. So we'll go through each of these factors just to give you an idea of what's involved during the development of the soil. Now, first, let's look at a soil profile. This is a very hypothetical soil profile, and this is used in that diagnosis as to what kind of a soil you have. And generally, the profile is a vertical slab of soil, and some of the older soils will show these layering effects, which are called or due to horizons. So the A horizon that you see here, the upper one um, in that column, it contains most of the organic matter, and it has the texture on which the soil is defined. So if it's a silt loam soil, this upper layer of soil, that's where the silt loam texture is. Most of the organic matter there is. And then over time, and we're talking of tens of thousands of years, these layers develop. That next layer is called the E horizon. That is usually a horizon where a lot of things, a lot of clays and nutrients have been washed out over time, especially in a fairly humid region where the soil water percolates through the soil, and that will give you sometimes a white layer, which is because it shows nothing but clay is left. Then you go into this lower horizon, which is the B horizon. This is where a lot of accumulated materials, such as clays, some of the iron oxides and chemicals uh, are deposited. So that is the idea of a soil profile. It's very simple, very quick, um, but usually you find an A, the upper layer, an E, the middle lighter layer, and then a B as a typical soil profile. All right, so two of the soil forming factors are parent materials and climate. I'm showing you here two very contrasting soil profiles. If you look on the left side, we see a what's called a Menfro silt loam, which by the way is actual the Missouri state soil. So it's a little bit of trivia that you learn here today. The Menfro is found on the river bluffs of both the Missouri and Mississippi River. It is the parent material was loess, which was wind-blown silt material that came through generally from a westerly direction and is overlaying on sometimes the glacial till that was left over after the glaciers had come down through the northern part of the state. And this soil has developed over time under a temperate, humid climate. You can see there's a lot of organic matter in this upper layer to about 1 foot. I believe that's in feet, and where the arrow is depicting. Silt loam's a very um, well aggregated soil, and the lower layer, about where the E or the B, I'm sorry, the E horizon is, at that second arrow under the 2 foot mark, shows where a lot of this organic matter and other material have been washed out. So it's a little bit lighter there. So this is a kind of a typical type of soil that you would see in the temperate region of the Midwestern part of the United States. The other profile showing on the right-hand side is what's called an oxisol. This was in a pit down in Brazil, and I visited there some years ago. I think it was in a coffee plantation. But the most noticeable feature is the reddish color of this soil. So this developed in residual clay under very high temperatures, high rainfall. So you have a lot of oxidation going on. This is all iron oxides that developed in here in a tropical climate. So you would find these soils in the tropics in the United States. You'd find something like this in Hawaii. Um, it has a very low fertility. Any phosphorus that is in there is tied up in these oxidized oxides such as iron and aluminum oxide. So just give you a little bit different. Obviously, what does that tell you? You would manage these soils much differently. I think that's obvious from seeing the contrast here. And in fact, we can look at the soil regions of Missouri and see that there are different types of soil around the state. You begin the boot heel down here, southeastern part of the state, lower right-hand portion of this map. Primarily, the parent material was alluvium. Material that washed down the Missouri Mississippi River was deposited in this huge delta area. So soils, the topography is flat. So it's a very deep soil, very fertile because washing down with the alluvium is a lot of phosphates, apatite type materials from let's say the Rocky Mountains, made it all the way there. So it's very fertile. Up here in the northeastern part of the state, we have these clay pan soils, windblown soils that has developed under a lot of water. Rainfall gives you the clay accumulation in the E horizon, and then in the western, northwestern, and this western Luss region, where situated where Kansas City, Prairie Birthday Farm, and on up northwest towards Nebraska, very deep Luss soils that I just showed you with the Menfro. Very fertile, deep, but they are on a rolling topography. They're on the river hills, which requires a different management. And then in the western part of the state, you have the prairies. These soils developed under prairie, under a little bit of a drier climate, but then again, they're have a lot of organic matter in them, have a lot of nutrients in them as well. They're on flat prairies, good organic matter, good well drained soils, and this would carry over into Kansas to some distance as well. So living organisms help form that soil. Remember those dark features in the Menfro soils. A lot of that's due to the extensive root systems of many of the native prairie plants that provide organic substances that also supported the microbial processes. And this is happening over millennia. Uh, One of the features of these plants is that as they grow, as they're photosynthesizing, trapping carbon dioxide out of the atmosphere for photosynthesis to form sugars, a lot of some of that trapped carbon is being released into the soil through a process we call rhizodeposition. And that is basically carbon fixed by way of photosynthesis is released into the soil by roots. And there's estimates anywhere from 20 to 60% of the carbon dioxide that is fixed by photosynthesis makes its way through the plant roots and is released into the soil. And so here, this inset down here in the lower right, you see this is a root surface of these plants. And what you see on here, these little structures are organisms that are attached to the roots because they can get that carbon substrate as it's being released through the root and metabolize and break down the roots. The roots that are sloughed off of the root system here contribute to the organic matter, contribute to the nutrition of these plants. So you can see in the diagram here of the plants, some of these root systems of Indian grass, big blue stem, switchgrass can go down to as far as 3 feet or more and really enrich this soil, giving it that dark color. And what else is it doing? Giving it good structure and aggregation. So very important to keep living roots on the soil. And that goes into these prairie ecosystems that we see here, whether it's a native prairie or a reconstructed prairie that's shown on the left half here. Support supports these. Sorry about that. Supports these systems um, in the soil. These soil food webs with these organisms. Again, some of the differences, we've seen this before, the deep prairie roots here on the left from Indian grass. This is on the same landscape, and right next to it, we have corn roots planted there. And you see the roots are doing very poorly. They do not go past um, hardly um, uh, 10 to 20 centimeters, whereas the roots here go down to 3 feet in the Indian grass. So again, um, some of the things that we're doing for cropping in our soils are not proper. These deep roots are being recognized out in the public, as you can see here, a label of a wine that show that the deep rooting habit helped the quality of this particular wine that it's depicted here. I think that's very interesting. Um, Topography is a very important factor. Up here on the summit, on the left-hand side of this graph, soils are completely different than what you find downslope. Down on the right hand, there's the foot slope, toe slope. Vegetation is different, so that's an important factor. And finally, time. We go from the left hand here, the soil may be very nondescript as we show on the very left profile, but over time, with the climate, interacting with climate, vegetation, landscape, you will see that these um, profiles develop with the horizons. So sometimes when we see a very recent soil, such as you would find in the Missouri or Mississippi river bottoms, they would be very recent because they have hardly any profile development, whereas on some of these soils in the Ozarks under forested situations have horizons and they would be a much older soil. So I mentioned a little bit about soil taxonomy earlier. Um, there is a classification system. It's based on the horizons in the soil, and I'm not going to get into it because it's a very complicated um, terms that are used to come up with these soils. But I just want you to be aware of that and the fact that we do have 12 orders of soil taxonomy based on the USDA classification system. In the upper left hand here or most of the soils in our region are what's called alpha soils, and these are soils that are fairly old, developed under temperate climates with fairly good rainfall, and they can be very productive, but they need some good management because some of these horizons may contain a lot of clay. Go down to the lower left. This is where the mollisols are called mollisols. These are the deep prairie soils. You can see by the black color, very fertile, um, good structure, have a lot of microbial activity, high organic matter. And then you can see next to it the oxysol, which we have discussed, low fertility, highly oxidized, and highly weathered due to the high precipitation and high temperature. Okay, so I want to get into a little bit of soil biology because that's really what my main interest is. Just to mention that with knowing the soil properties, we understand that the soil biological activity must be conserved, and then soil management or in other words, human activities, which we must consider ourselves as a living organism, is a key to soil health. And so all in this upper left-hand box, you can see our management can lead to a lot of problems if we overdo it with tillage, run over with these huge pieces of equipment on farms. It can compact the soil, reduce that porosity, residue management. Some guys are harvesting corn vegetation after harvest, removing that residue, which is the source of organic matter. That really needs to be managed. And it affects all this biological activity and also some of these soil properties, aeration, organic matter, the amount of nutrients in the soil that is necessary for this block of soil over here, a hypothetical soil food web that you see on the right component of that block of soil that has to support that diversity. So the influence of management practices is critical because it does affect the biological component and its interactions. All right, just a little introduction into soil biology. Um, as it is a main component contributing to soil health, which we'll get into the next lecture, you have to look, you can look at the diversity, abundance, and distribution of organisms in the soil. And one of the things we don't want to forget are the terrestrial arthropods, the insects above the soil, because these are really taking a hit these days due to climate change, the amount of pesticides are being released on agricultural soils, and they do have a great impact on the diversity of the organisms that I'm depicting in this triangle. So if we can characterize the microbes in the soil on a biomass basis or a numbers basis. So at the very top, we have those that are least in numbers, such as the earthworms, some of the insect larvae, the other arthropod types, different types of worms. They are low in numbers, but they contribute to highest biomass, which is the macrofauna here. Then we have, okay, are we about?
Just a quick time check for you.
Okay, 5 minutes.
That sounds perfect.
Okay, so look in this next level, we have the other faunal components, the nematodes, a lot of the mites, the small collembola type insects. These would be mesofauna. Then we have the microfauna, which include the protozoa, the smaller nematodes, these kinds of organisms. And then the microflora. This is probably the most important section of the biological component because the fungi, the bacteria, the archaea, and the actinobacteria, they're the ones that drive the metabolic and biochemical forces. They're the only ones that really can degrade these components into simpler parts and mineralize these nutrients for plant uptake. And then the lowest tier here, which are the viruses. Believe it or not, we have a lot of viruses in soil. We call these phages, and they are the most numerous. There may be up to 1 trillion or more viral particles in the soil. These are what I would call the natural nanoparticles because they are in billionths, whereas up here, these microflora are in the millionths of size ranges. They're very important because they can control the populations or the abundance of the microflora, but they can also transfer genetic material. There are the natural genetic engineers in the soil. They can transfer genetic material from one infected bacterium to another. Let's say if it has a gene coding for a particular pesticide breakdown. So just be aware of that. They are the most numerous, and of course, they're the least. They are the least um, uh, in biomass. Let's see. I lost my. Um, let's see. Let me zoom in a bit here. Maybe no. Let's see. I've lost my. Oh, here it is. I'm sorry. I'm new to Chrome. Okay, some of these organisms, this is what they look like. Real quickly, we'll get into this again, but this gives you an idea of the microbial diversity, the consortia we mentioned about processing organic matter that is needed. But we have an example of structural diversity, the bacteria here. Actinobacteria, basically bacteria that have filaments like a fungus. The nematode, the worm structure here. The fungi down the lower left, the filamentous forms that you see, the algae in the lower right are very important um, because they can photosynthesize and move carbon right into the soil. Then we have the macrofauna in this right-hand side, the earthworms, and up here I have the bacteriophages, the viruses, and this is at a billionth of a meter or 10 billionth. And you see some of them look very strange, almost like a landing gear, landing satellite on the moon. But that's how they move their genetic material around. In that second inset there, they can actually pierce a needle-like projection into bacteria, insert their genetic material, and reproduce, just like a human virus as well. And these are some other depictions of the soil biology. Uh, if you look at that top, we can go all the way to the nematode feeding structure. The last one in the corner there is a water bear. Beginning in that lower second section, we begin with collembola, different types of millipedes and worms. The last one is a collembola, springtails. In the lower panel, beginning at the left hand, you have earthworms and going all the way to a mole. So all these are very important. I will show that in the soil food web. They contribute, they interact, they support the biology in the soil, especially the microfauna. And they can be depicted differently. And, well, this picture really shows you the variation in the distribution of these microbes. If you begin on that right side, the rhizosphere of the root is, you have a lot more diversity, a lot more different organisms there. Then you get into the detritosphere, which is old organic matter. You still have some. You have the yellow strand there, which are the fungi going through there. But then when you get out into the bulk soil where there's root-free, the abundance and distribution and diversity is quite a bit lower. Okay, we've come to the end of this particular presentation, and Hunter, I'll turn it over to you if there's any questions. I'd be glad to try to answer these.
Thank you, Dr. Kremer. That was a great presentation. It's so cool to think of the alien world that's below us all the time that we so rarely take the time to consider. I know I was outside the other day, noticed a few cracks in my yard because it's been so dry, and I was wondering if that's because it's a poor quality soil or if that's purely because it's been so dry, and that will work itself out eventually.
I'd say it's a little bit of both. Depending on if it was in your lawn, or it kind of depends on what the vegetation is there as well. Especially, let's say in our area, we can find a lot of cracks if there's a lot of clay in the soil, because clays, they go from one extreme to another. They can swell, or when it gets very dry, they will pull apart and they can crack the soil. So if you have a lot of clay in there and the water is deficient, you will find that. Also, if you have a lower part of organic matter and not a lot of root depth, and it's dry like this, that will eventually lead to that as well. I remember in the drought of 2012, we were very concerned because there was a long period of time where we just didn't get any rainfall. It was very hot. And I took a sample out in one of the field plots, and it was, like less than 5% moisture. It was very dry. And you would think that a lot of these microbes would die under a situation like that. But as soon as moisture was returned from the remnant hurricane that we got through here, it all came back to life. I think one of the things we forget is that also, even though they depend on an aqueous environment, the substrates, the carbon that they use, also needs to be in a liquid form. So everything just kind of shuts down at that point. The carbon is there; it's not going away. It's just dry. So there's a lot of factors that go into that situation. I appreciate the question.
Oh, certainly. I appreciate the answer. I know we have another question that is, what can be done to increase the quality of the soil in native plant beds at individual residences?
Yes, good question, and I'll address that in the next lecture. But basically, um, anything that you can do to improve plant growth through, let's say, compost additions, can help that. Because that will improve not only organic matter, it will also feed the microorganisms that you have there. Um, Obviously, keeping it well watered. Although a lot of the native plants developed under this condition and only need a lot less water than, say, your typical bluegrass or fescue lawns. So that is that's an opportunity to maintain a cover on the soil. Um, there are also some biological preparations that I've looked at, a lot of what's called biostimulants and these kinds of things. Biological preparations that can be used, especially depending on if your native plant was established in, let's say, a renovated landscape, if it was an old lawn or a plant bed or something. It's good to check to see if you have an organism called the mycorrhizae, and it's such a mycorrhizae fungus that associate with the roots of these plants, and it really helps them take up water, take up nutrients. In fact, it will go out in trade for carbon from the plant. It will access nutrients in the soil, especially phosphorus, some nitrogen, potassium, that kind of thing, and bring it to the plant. That will improve productivity. And you can find these mycorrhizal inoculants even at some hardware stores. And so if you think if there's a question about that, I would recommend adding those kinds of organisms just to be sure that you have the best root growth that you can get out of that particular bed.
That's a perfect setup for the next presentation you'll be doing today.
Right.
I'd like to thank everybody for joining us. Make sure to please take a moment to fill out the session evaluation and let us know what you thought. Our next sessions are starting at 3 o'clock today. While you wait, please visit our exhibitor chats or check out our generous sponsors. If you have more questions for Dr. Kremer, please join us at his office hours following his second presentation at 4 o'clock today. The office hours are at 4. And if you want to listen to the session again or catch one that you missed, Planet Native 2022 is recording all sessions, and you'll have access to the recordings for 1 year to watch as much as you'd like. Thank you, Dr. Kremer, and thank you, everybody, for joining us.
Okay, thank you, Hunter.